Good evening, ma'am. Good evening, Ajay.
Good evening, sir.
Good evening, Ganna.
Good evening, sir.
Good evening, ma'am. Good evening. Good evening, Ajay.
Good evening, sir.
Shall we start now?
Yes, sir, you can start, sir.
Yes, ma'am. Thank you. Thank you so much, ma'am. Good evening, my dear students. So, once again, you all are welcome to the another biological session, part 6. Part 5 already we discussed. What is by immunoglobulin? What are the types of immunoglobulin? How it will be neutralized? All those things already we discussed, isn't it? Today, we are going to discuss the another part of human welfare, continuity. Okay, what are they? See here. The first we are going to discuss the type of acute immunity. What is by acute immunity? Already we discussed innate and acute. Okay, so innate, what are the things already we discussed? Already you know a little about. And acute immunity is also we are going to discuss. In this category, you must note it down, the acute immunity. First of all, what is meant by acute immunity? What are the types of acute immunity and how it will be activated in the human immunological system? We are going to discuss one by one. First of all, acute immunity. The acute immunity is immunity you develop over your lifetime. This is up to the lifetime fully. Such type of immunity is called as, what is that? Acute immunity. Develop over your lifetime. It can come from a vaccine exposure to an infection or disease. Suppose any one of the foreign particle enter in your body, what happened? Automatically, we are exposing the vaccination, isn't it? So that vaccine, what happened? Vaccine activate upon the disease, fight against many diseases. That condition is called as acute immune. See, another person of the antibodies, the disease also, the vaccination is also obtained from the another person's antibody. Okay, such type of immunity is called as, what is that? Acute immunity. Most of the cases, the acute immunity may be obtained from the animals like, animals like, which one you know? Take an example, horse, rabbit, chimpanzee, monkey, and these are, and finally the human beings also. They were obtained the antibodies. It can also be classified into following. Already it was told. The acute immunity is also further divided into two types. One is called as active acute immunity and natural active immunity. So these are the two types have been reported. Once again, repeat for you. What is by acute immunity? Acute immunity is an immunity you develop over your lifetime. So it can come from where, the vaccine exposes to an infection or disease, another person's of antibodies. It can also be classified into the following types: active immunity, natural active immunity. See here one by one. See, active immunity. First one is called as active immunity. Okay, the active immunity, what is that? When a host is exposed to an antigen is nothing but when the foreign particle enter in your body, the foreign particle, bacteria, virus, or any other protozoa enter in your body, automatically the protein molecule secreted. That protein substances is called as antigen. Okay, when the antigen produces, automatically the hosts also produce the antibody. That is very, very important. See here, when a host is exposed to the antigen, antibodies are produced in the host body, isn't it? This type of immunity is called as active immunity. When the foreign particle enter, immediately responds, that is called as active immunity. Okay, don't forget it. But the active immunity is a slow. It is not rapid in nature. It's not fastly it happen. It will not happen. Okay, it is a slow process. And the active immunity is also further divided into natural and artificial. Natural active immunity and also artificial active immunity. Once again, active immunity comes under the heading is known as which one you know? The acute immunity. Okay, the acute immunity divided into two types. One is called as active immunity, another one is called as natural active immunity. The active immunity also further divided into natural and artificial. Okay, what is mean natural? We will see one by one. Naturally means in our body. The natural active immunity. The natural active immunity is acquired when antigen gain access to the body during natural infection. Natural infection. What are the natural infection mean? Just imagine. See here, the smallpox, measles, mumps, and these are all are comes under the heading known as natural active immunity. When this type of microorganism, taken a smallpox also caused by which one of the microorganism? Will you please? Measles, mumps. What are the microorganisms? These are all are once attacked the human body. During that time, automatically what happened? Naturally, the body produced which one you know? Antibodies. Okay, so this is called as which one? A natural active immunity. Once again, repeat for you. Natural active immunity is acquired when antigen gain access to the body during natural infection. What are the natural infection? Smallpox, measles, mumps, and natural activities. Okay, see that. And next one is called as artificial active immunity. Artificial active immunity. See here, the artificial active immunity is the resistance induced by vaccine. It is also an outer source. Here, naturally present in it. Isn't it? It's already present in our body. But this in this category, see that one, it is a vaccine is also introduced from the outside. See here, the vaccine, how it will be? See, the vaccines are preparation of live or killed microorganism already in our body. There are two types of immunity. Natural immunity, naturally produce the antibody, isn't it? In our body having an immune system. Artificial, some of them are, they do not fight with the natural immunity. They will not fight with the natural antibodies. Some of the antibody produced in your body, it will not fight the, which one, you know, some diseases. That time what will happen, you know, you have to move on to the doctors. So they analyzed the diseases and come to the conclusion. So they may provide, they may recommend, they may, what is that? Prescribed a particular vaccine that or there are products used for the immunization. Okay, so that things. Take an example, COVID. We were injected by the government, isn't it? Indian government. How many vaccines? There are 3 doses. The first one is primary and secondary, and third one is called as precautions. These are the type of vaccines we were used. Okay, so here live and kill, that vaccines are prepared by the microorganism from the same microorganism, isn't it? So they may be killed one, or sometimes they may be used as a live, and some of them are attenuated. That some of the modification also takes place. Such type of, what is that? Components are called as vaccination. They have the ability to fight against many diseases. So they are all are called as, what is that? Vaccine, antibiotics. Okay. See, they have given so many examples. See here, first, for example, live BCG. Note it down. This is very, very important. Live BCG. Yeah.
So what does BCG stand for, sir?
Pardon?
So
BCG.
what does BCG stand for, sir? Full form of BCG, sir.
BCG. Yes, I will inform. I will tell you. Okay, so I will tell you later. Okay, right. Say BCG, Bacillus, I think it comes vaccine for tuberculosis. Killed tap vaccine for enteric fever. A live Sabin vaccine for poliomyelitis. Already we discussed Sabin and Salk, isn't it? Poliomyelitis. And MMR, already we discussed it. Mumps, measles, and rubella. Okay, see here given. And the killed sacs. Here we must note it on which one is live, which one is killed. That is the ratio. Okay, the live vaccine for BCG, killed tap, live Sabin, and MMR vaccines for measles, and the killed Salk vaccines. See, so these are the vaccines given for which one, artificial oriented, artificial active immunity that can be prepared from the outside. See here, what is by BCG? I will tell you. Okay, see here. I will tell you one by one. Okay, see here. Already we discussed that things. BCG, Bacillus, and coumarin, like that it comes. Okay, so I will tell you one by one. Again, I will tell you, Ajay. Okay. See, now attenuated vaccine, how they are preparing. This is the methodology. Okay, this is the methodology for the preparation of a bacteria or virus, whatever it may be, attenuated, repeated passing in the cell. Again and again, they will give. As a result, what happened? A culture or animal embryos, chick embryos, maximum they were used for the live attenuated vaccine preparation. They were used chick embryo only or animal embryos. Okay. And then how it will be, see the mutation, a weakening. Okay. How many times they may take for to how many years? 10 years or less than or more than 10 years. Transforming of wild virus into the vaccine virus. Okay. Live attenuated. Live means living microorganism that may be injected into the sick. Okay. Embryo or animal embryo. Injection of the small dose of the vaccine virus or bacteria. Vaccination. Replication of vaccine virus or bacteria in the host again and again. What will happen? More than 10 times, 5 times, like that they may induced. So injected. As a result, specific immune response. How it will be automatically? What happened on the vaccination? Injected mean automatically the specific immune response will be reflected from the body. Induction of an immune response. Induction of immune response. Again, what happened? Finally, it protects against the real infection. This is the primitive steps taken by the and research oriented. They were taken as a what is the analytical aspect, otherwise called as vaccine protection. So before the vaccination, these are the steps were utilized by the scientists or used by the scientists. Then only the protection against the real infection activity will be find out. Okay, so likewise, already we know, but there are so many scientists discovered that which one or introduced the vaccination for COVID, isn't it? First one is called as primary. They may prepare from the outermost coat, the protein molecules, otherwise called as that is a hooks. Okay, and sometimes a inner part of the cell wall like that, and sometimes they may be used as a which one the DNA and RNA molecules. So these are the which one steps are used by the scientists in commercial preparation of and vaccinations. So next, see that. So this is BCG. Okay, I will tell you what is my BCG. Okay, Bacillus and calamitous gone like that it comes. Okay, so this is the person. So BCG vaccine when we are giving, automatically the body will be injected side look like this one. So here there is a dark like cell. These are the neutrophil cells, otherwise called as macrophages. Okay, the dendritic cells also activated. See that the dendritic cells. This is called as dendritic cells react with the macrophages. As a result, the cell wall skeleton will be like this. The cell wall skeleton will be like this. Structure will be like this. It will be converted into this form. And also automatically what happened in MTB, these are all the activated macrophages. When the immunization activated macrophages, it look like this one. These are all are a receptor. The MR2, MR4, TLR9 like that. There are so many receptors also present in it on the cell surface. Okay, this is the one step. Another type, what happened automatically when it injects, automatically it enter into the lymph node, lymphoid organs. Okay. As a result, it is also there are same process occurs, and also the surface they are having the receptor cells. It bind with this one, automatically they produce MTB. And another category, this is the another steps. So here, finally, what happened? The specific antibodies are produced. This is also not an antibody. This is the inducing of the antibody, inducing the steps, inducing of the chemical substances of the antibody. These are the cells. These are the cells participate the specific antibodies. Specific antibodies react with that. These are the steps. These are all you will study in the where when you are going to study the doctorate in the first year or second year or third year. Okay, immunology is one of the part. So that time you can study deeply how it will be, what is by dendrite cells, what is by macrophages, activated macrophages, how it will be infect the diseases like that. Okay, so these are all obtained from the Google. So okay, so no need to worry, just note it down how it will be acted. So likewise the same is that, see here it is also introduces sporocytes. Okay, this is the diseases. Okay, here we are introducing the injections, BCG. How it will be reacted? Already in order there is a receptor is the joined with them. So as a result, what happened automatically? It produces some chemical substances. That chemical substances automatically lyses the RBC cells. Finally, it will be eliminated like that. Normal tropocytes. So these are the steps. Okay, how it will be acted like that they may told. Okay, go to the next one. See, passive immunity. This one is called as natural. Second one is called as passive immunity. The passive immunity is also another type of immunity, acute immunity. So it is ready-made antibiotics are directly given. This is called as, what is that? Passive immunity. Passive immunity is provided when a person is given antibiotics to the disease rather than producing them through his or her own immune system. The own immune system is not properly, what is that? Fight against the particular disease or the invading of microorganism. What will happen? We will provide directly antibiotics directly giving. Okay, that one is, what is that? The antibody inducing substance, that is called as vaccine, that induce the immune cells and produce the antibody. It's an indirect method. This one is called as directly we are giving. So when the ready-made antibiotics are directly given to protect the body against foreign agent, it is called as passive immunity. Okay, so passive immunity is directly involved. Okay, fight against the diseases, particularly the foreign particles, foreign agent. Okay, whether microorganisms such as a virus, bacteria, and protozoa, whatever it may be, such type of is called as passive immunity. Once again, repeat for you. Here you must note it on passive immunity, ready-made antibiotics. We are giving directly. What will be the action here? See here, ready-made antibodies are directly given to protect the body against foreign agent. It is called as passive immunity. When his or her own immune system, they will not produce the antibodies. That time we will introduce the antibody directly. That is very, very important thing. See that. So the passive immunity is also further divided into two types: natural passive immunity and artificial passive immunity. Acute also two things. Okay, natural and artificial. So here the passive immunity is also divided into natural and artificial. Here natural passive immunity. What does it mean by natural passive immunity? See here, students. Natural passive immunity is the resistant passively transferred from the mother to fetus through placenta. Naturally, what is that? The resistant power transfer from mother to fetus through placenta or umbilical cord. So such type of immunity is called as natural passive immunity. Okay. See, immunoglobulin G antibodies can cross placenta. They may ask the question sometimes, which one of the following? Okay, immunoglobulin can cross the placenta. Like that, they may ask the question. That time you have to tell immunoglobulin G can cross the placenta. After birth, immunoglobulins are passed to the newborn baby through mother milk, otherwise called as colostrum. Colostrum is nothing but already in our it is nothing but the first mother milk. It's a pale yellow color. Yeah.
So, but colostrum has IgA antibodies, right, sir?
Yeah. It's passed only. Okay, which one is the high concentration? They may ask means you have to tell immunoglobulin A. Okay, but after the birth, the immunoglobulin G are passed to the newborn baby through mother milk. So it is nothing but the passing way from the milk. Highest concentration of milk. Okay, in mother milk, which one is vitamin? Immunoglobulin A will be very high in nature compared to immunoglobulin G. But immunoglobulin G can pass through the mother to fetus through mother milk. That is the reason. So colostrum is the first mother milk. It is in pale yellow color. The colostrum contains high amount of immunoglobulin G. This is very, very important. Okay, once again repeat for you. The natural passive immunity is the resistant passively transfer. Resistant passively transfer means what is that meaning? Immunoglobulin G. It is antibiotics. Okay, passed through which one through the placenta. Okay, so immunoglobulin G antibodies can cross placenta already after the birth. Immunoglobulin G are passed to the newborn baby through mother milk. So we come to the conclusion: colostrum contain high amount of immunoglobulin G. Which one is highly passed? Means which one immunoglobulin A. Immunoglobulin A is high concentration. Once again repeat for you. Immunoglobulin G is a what? Passed to the newborn baby through mother milk. Okay, immunoglobulin G is highly passed. That's why color will be very yellow in color or pale yellow in color. But immunoglobulin A is how much? High concentration, highly rich concentration in mother milk. So we come to the conclusion G and A is present in the milk. Particularly immunoglobulin G is passing through the milk, but immunoglobulin A is high rich content. Like that, you come to the conclusion. Okay. Suppose if you have any doubt, we will clarify it next classes, isn't it? See that colostrum. That's the first milk. See that a pale yellow color. This is highly, what is that? High content of immunoglobulin G. Okay, but the mother milk, second milk contain immunoglobulin A. So these are the two things is that the colostrum containing immunoglobulin G, the normal milk containing immunoglobulin A is a high rich content. See that. Colostrum is a rich in immunoglobulin A antibody. Already told you. Okay. So that is very, very important. So many times they were asked the questions. Colostrum containing which one of the following? High. Like that they may ask the immunoglobulin A. Okay. The mother milk contain antibodies that protect the infant properly by age of 3 months. So that's why the mother milk provide how many months? 6 to 9 months. Sometimes 9 months or 1 year. Like that they may give. Okay, these are the very, very important. So many times they were asked the questions. Mother milk content, which one is the highest concentration of the antibiotics or immunoglobulin? Which one is easily cross the placenta? Like that they may ask the questions. So you have to differentiate between these two. It's very, very important. See. And artificial passive immunity. Next one. The artificial passive immunity resistant passively transferred to the recipient by administration of the antibodies. Recipient. Okay, recipient means those who are receiving. Those who are receiving from the administration. Those who are giving. Or donor as well as a recipient. So this is done by hyperimmune sera of animals. Already told you. Okay. And human serum containing antibiotics. Some of the animals also has the similar, which one antibodies like as a human being. That serum is also isolated from, that means antibodies also isolated from the particular animals and also injected to the human body. Fight against many diseases. Okay. So anti-serum, that is called as anti-serum. The anti-serum is prepared in horses. Here only one has given, not only this one, horse, rabbit, chimpanzee, and also sometimes they may use it in the human beings also. You have to check it. All the mammals accept these type of antibiotics. Means what is that? It can be passed on to the next human beings. And also the age wise also that. Okay, 6 months, 10 months, fetus, 1 year, 2 years, 3 years, and 5 years, up to 5 years. Then 5 to 10 years, 10 to 15 years, 15 to 30 years, 35 to 45 years, and also adult, old person, 60 years, and then pregnancy women. So you have to, they are having so many slides. Okay, all are analyzed and come to the conclusion, then only introduce the market or a commercial purpose in the market. Anti-serum is prepared in the horse by active immunization of the horses. Active immunizations. We are giving the immune to the horse. What will happen? Automatically they induce the cell inside the horse immune system and produce the antibody. Again, derived, and also they can be produced for commercially more number. That is the reason. Horses with the tetanus toxoid, bleeding the serum. These are all are prepared from the horse. Okay, tetanus toxoid, bleeding the serum. These are the two things. See here, the artificial. See that serum is used for the passive immunization against tetanus. Similarly, when you are preparing the horse, when you are introducing the tetanus microorganism to the horse, so what will happen? Automatically they produce the immunity. Tetanus anti-serum produced against the tetanus. Suppose we are introduce the rabies, it is produce the anti-serum of rabies. Suppose we are introduce the diphtheria immunization. So what will happen? Automatically they will produce anti-diphtheria serum. Suppose it is a gangrene serum, ACGS, anti-gas gangrene serum. This also prepared by the immunization against the. Okay, these are the anti-serum. These are all are called the anti-serum. They are all are having the antibodies fight against the specific disease. For example, rabies diseases, you have to provide anti-rabies serum. Diphtheria, anti-diphtheria serum. So anti-gas gangrene serum is also this disease. Tetanus. These are all are prepared from the animals or artificially introduced passive immunization. Then only. So this is also okay. See that tetanus toxoid. Okay, we are giving. As a result, what happened? Immunized or this is considered as a immunized or when you are giving the orange color, that means the medicine vaccination given, immunization given, the body fully what happened, changed, So immunologically, what happened? The response as a result of production of antibodies within the coast, isn't it? Horse, whatever it may be. Again, we may collect from the which one, immune horse serum. You have to collect from the blood from this which one? Horse. After that, you will get the tetanus immune globulin. From this one, tetanus immune globulin is there. So we have to pass on to the next horse, unprotected one. It may dead, maybe possibility. Okay. Again, suppose you take from this one to again, and I'll pass on to the next one, they can protect it. So these are the steps. These are the steps. First, you have to introduce the immunization for the particular horse, collect the sample, and I'll introduce the another one, and introduce the another one. This test will be going on up to 100, 10, 200,000 like that. Okay. Suppose when you are introducing from the first to first horse, second step. Okay, the horse may be dying. So the process, what happened, they may find the fault. What it happened, why it happened, like that they may come to the conclusion. Some of the modification also done by this particular vaccination. After that injection, suppose if it is alive, okay, it transfer from the another horse. So it may be protected. Likewise, the immunization is also prepared. That is the step. See, the same things is that. Sometimes they were used the spike of the protein. Okay, you know about that. There are so many biomolecules present in the viruses, isn't it? The outermost protein coat is called as what is that? What is that? Protein, called glycoprotein. Take an example of virus HIV. Glycoprotein 121 and glycoprotein 47, like that it has. That protein molecules taken from or isolated from the viruses or pathogen, it can be introduced into the, which one, horse. So automatically that thing's also done in COVID cases. COVID cases already in our world, but we are all received the vaccine. We also put the vaccination, isn't it? That vaccination contain which one, which one of the molecules of the pathogen. It may be spike protein, or sometimes it may be a cell wall protein. Sometimes it be a nucleic acid, a particular portion. Okay, that is the key component for the production of the vaccinations. Here they have took which one, spike. That means the outermost layer, there is a spike protein. The spike protein introduced by the horse. What happened, the particular period, that is called as inoculation. The symptom will be comes out, isn't it? After that, it will produce automatically the horse response to the horse immunity, immune organs responsible for the particular spike protein, that is foreign protein, otherwise called as invaded foreign molecules. As a result, what happened, the antibodies are isolated from and also produced for that SARS viruses, and also neutralized and introduced to the another, which one, So these are the steps were used for the preparation of the vaccination immunization. See, the same step is here, okay, but the protein molecules is not clearly visible. So this is a step. Is just imagine this is the virus. Introduction takes place, collection of samples, and also multiplication. For so many multiplication also done, and also we will get the antibody. The antibody is used for the commercial purpose. Okay, see that, and we will pass on to the next one. Is before that one, primary and secondary immune response. What is by primary immune response means? Just imagine first a particular microorganisms enter your body or exposed to your body. It produces the first antigen, that is called as first exposure to an antigen. See here, the primary immune response occurs after the first exposure to an antigen. When the first affected by a particular virus or bacteria enter in our body, that is the first time. Means the first time what happened? Produce the antigens. Such antigens are called as first exposure antigen. Suppose if it is again it enter, that is called as secondary immune response. The primary immune response result in the generation of memory immune cells. Suppose the first exposure an antigen, first time enter. After that, the second time it enters means the first antigen it's act as a memory immune cells. Okay, see here, a second immune response occurs as a result of second exposure to an antigen. Once again, it repeated. Again, and I repeat for you, primary and secondary immune response. When the foreign particle first time enter, automatically the first exposure to an antigen also produced. Okay, suppose after sometimes what happened, again the same microorganism, same pathogen enter in your body. It's a second time. Second time means what happened? The first time already exposed to an antigens also have the memory cell. It induce it. That's why it is called as memory immune cell. It acts against the secondary immune response. Okay, you got it this point? Ajay, can you clear these points? Ajay?
Yes, sir.
Oh, yeah. So for the next one. So this is a diagrammatical representation given, present the primary immune response. Okay, see here, macrophages activate. So first time what will happen, killer cell activated by the killer cell as well as the other cells also, B lymphocytes also. They produce, these are the memory cells. Okay, again it will repeated, the same cells also, again it's also repeated. What happened? It act upon it, as a result, what happened, completely destroys. This is the things. See the first invaded T lymphocytes and killer cells activated by the B cells, B cells, T cells, key cells also activated. As a result, first specific antibody against the antigens, it will be produced. Okay, the plasma cells produce specific antibody for the particular invading microorganisms. This is also stored in the immune, that is called as memory cells. Again, the same microorganism enter here, automatically the memory cells act upon, it will be destroy the things. So the antigen antibody reaction takes place, and macrophages also takes. Macrophages, otherwise called as what is it? Phagocytosis, cell eating. So the immunity automatically enter it and completely neutralize it. And also we already studied neutralize the pathogenic activities. As a result, it will be recovered. Okay. So remaining, it will be stored in the form of memory. Okay. Remaining in things stored in the form of memory. Again, the third time it enters, what will happen automatically? The secondary immune response will also activate. The first memory response and second memory response also activated upon it. As a result, it will be neutralize the pathogens. See. So this is the initial exposure of antigens. First time, what will happen, the primary immune response, how it will be very less in nature. Very less in it. This is the maximum activity. The antibody concentration. The antibody concentration, how much when we are giving, this is the concentration. This is the 5, 10, 20, 50, 55, like that. Say 5, 10, 15, 20, 25, 30. You just imagine 13 is the maximum responsibility. Okay. But same level here, second time when we are giving, what happened? This is the stage. Okay. See, up to the level, this level. Here, where is it? Here, where is it? Just look at it. You can see the differences between the first immune response and the second immune response. When you are giving the initial exposure to the antigen, this is called as primary exposure to the antigen. The response will be very less. Suppose again you are giving the second exposure to the antigen, the response will be very, very high in nature. So see here, this is the first step. This one is the double of the second response. This is the graphical representation of the antibody concentration against the immune response time and immune response also. And then we will pass on to the next one is vaccination and immunization. What is meant by vaccine? What is meant by vaccination? Who discovered the vaccination? And that things we are going to see. See, vaccine is a suspension of inactivated pathogen or antigenic protein of the pathogen. The antigenic protein, sometimes it may be a nucleic acid. We will discuss what are the molecule used for the immunization or vaccine preparation. We will see it once by year. This is very, very important for your future. Okay, don't forget it. Okay, when you are going to study doctor, opposite, you have to tell vaccination. The vaccination and immune. So vaccine is a suspension of the inactivated pathogen or antigenic protein of the pathogen, which is taken orally or injected to the provide immunity for the pathogen. Okay, sometimes they may be used for orally. Polio vaccine, oral polio vaccine, they have given orally. Sometimes it may be injected. Most of them are injected only, isn't it? That is called as injector vaccine. How they will prepare the inactive pathogen and also antigenic protein. Okay, see here, vaca is nothing but the cow. Okay, how they will prepare the vaccine preparation is live or killed one. Okay, the preparation of live or killed microorganism or their product used for the immunity. Already told you what is meant by product. The virus or bacteria cell wall is made up of a glycoprotein or protein molecules. The spikes also there. Sometimes they may be lipids. Okay, some of the bacteria, some of the virus also having the protein molecule that is called as capsid and capsomeres. Inner there is a nucleic molecule that sometimes DNA or RNA, but both it will not present the single pathogen, isn't it? Both are not present. See here, first who discovered the vaccinations? Edward Jenner in 1796 prepared the first vaccine, cowpox, a smallpox. You note it down. This is very, very important. Who is the father of vaccination? Edward Jenner. Okay, who discovered smallpox vaccination? or cowpox vaccination. It's the first one. Okay. And then second one is Louis Pasteur in the year 1879. See here, 789, isn't it? 1879. Find that the aging culture of cholera bacteria did not cause the disease in chicken and also made them immune of fowl cholera by refining the technique of attenuation. What happened in the Louis Pasteur once upon a time? What happened? No. Find out the another one, cholera bacteria, where it will not cause any disease in chicken, also made them immune of fowl cholera. So this is the another steps. Again, the Pasteur discovered 1885 developed a vaccine against rabies. Rabies vaccine also discovered by the name of the scientist Pasteur. The year will be differ. The year will be differ. Okay, note it down. This is very, very important. Louis Edward Jenner and Louis Pasteur and 1879, 1885. How many years difference between these two? 6 years difference. Cholera bacteria and against rabies vaccinations. And then see the Von Boring. Note it down. This is very, very one of the important scientists. Discovered the process of passive immunization and prepared the anti-diphtheria serum by the injecting diphtheria antigen into the sheep. Just to imagine, so Von Boring is identified or discovered the vaccination diphtheria. What is meant by discovering? What is meant by invention? What is the difference between the invention and the discover?
So,
Yeah.
so invention is when they make something on their own, sir. It's new, sir. Discovering is when it is already present, but we don't know of it, and then they find out that it exists.
Okay, very good, very good. Discover means newly introduced, newly identified, isn't it? Newly, isn't it? Discover. Invention means what is that? From that one, discover to invention. So already present in it. That one too, or some modification, alternative form, or were identified or found out. That is called as what is that? Invention. Okay. The Von Boring discovered means a new one. Or discovered means what is that meaning? The process of passive immunization and prepare the anti-diphtheria serum by injecting the diphtheria into the sheep. So this is called as discover. Von Boring is the father of passive immunization. Sometimes they may ask the question, who is the father of passive immunization? Means Von Boring. By the end of 1920, vaccine against tetanus, diphtheria, pertussis, and also tuberculosis become available. When it available, like that they may ask the questions. Okay, 1920 year, just note it down. Okay, and one more is that Von Boring, when he discovered these things, just imagine and also find out the answer. Also, Von Boring, which year he discovered the passive immunization. That's why he is also father of the passive immunizations. That things also just to find out. Actually, and then the first vaccine for cholera by Louis Pasteur, 1880. And also Emil Rox, the first vaccine for rabies by Louis Pasteur and Emil Rox, 1885. And in 1890, the first vaccine for tetanus serum antitoxin by Emil Von Boring. The 1890s, see the difference, 80, 85, 90, 96. Here 5, listen here, 5 years difference between these three. And this one is 6 years. First vaccine for typhoid fever by Almroth, Edward Wright, and Richard Pfeiffer, and also Wilhelm Kohl. These are the scientists discuss final stage. There are some development is also there in okay. So this is the types of vaccine: live attenuated vaccine, inactivated killed vaccine, toxoid vaccine, cellular fractions. Cellular fraction, any one of the microorganisms, they are having the cell wall or cell membrane, isn't it? From that component, any portion, fraction means it's a small portion of the cell wall or cell membrane. That is called a subunit. Okay, subunit, sometimes it may be a DNA or RNA molecules also. Conjugate vaccine. Conjugate means it may be connected with the same microorganism from the one to another one. They both are united together and produce a new one. And also acellular vaccine is also another one. DNA vaccine, recombinant. Recombinant means it's also related to DNA to DNA. Okay, one of the highly resistant DNA. Another one is also number of what is that? Another components. They may be united with the technology called recombinant technology. As a result, the vaccination may produce means that is called as recombinant vaccine. Combination vaccine. Combination. You have sometimes the two molecules also united together. Then only the vaccination will be active. Otherwise, it will be inactive nature. Suppose if it is in separate manner, it will not active properly. You have to add two combination vaccine. So these are the types of vaccine. See. So classification of vaccines also same times also given here. The types of vaccines here live attenuated bacterial BCG. Okay, the viral mumps, measles, rubella MMR, polio vaccine, polio vaccine. Killed one, say bacterial pertussis is a killed one. Viral polio vaccine is also IPV. Okay, that is also a killed one. Oral polio is a live. Note it down this time. Sometime they were asked there. Another one is called as biosynthetic vaccine. Biosynthetic means man-made substances that are very similar to species of the virus and bacteria. It is commercially prepared in the lab. That components will be similar to that of the infected pathogen or virus, bacteria or harmful viruses. Haemophilus influenza type of B vaccine also is a biosynthetic vaccine. This is commercially prepared in the laboratory. Toxoid vaccine contain the toxin or chemical made by a bacteria or virus. This is called as toxoid vaccine. Diphtheria and tetanus and also DTP. These are all comes under the getting known as toxoid vaccine. These are the another things. Sometimes they may ask the question, which one is a killed one, which one is a live one? Will you please, which one of the following is a biosynthetic vaccine? Which one is not the following is the not biosynthetic vaccine? Given examples of toxoid vaccine, like that they may ask the question. You have to remind this point. Very, very, very important things. Okay. So again, the same thing. Live, inactive, conjugated, and also DNA and mRNA vaccines, viral vector vaccines, and toxicoids. Okay. These are the things. See, here you must note it down. These are the vaccines. First one is called as RNA vaccine. RNA vaccine is not given there. They're giving the nucleic acid, particularly recombinant vaccines. Okay, combined vaccine like that they give, isn't it? See, RNA vaccine is also present in it. DNA vaccine is also given, and recombinant. Listen here, recombinant. Whether it may be a DNA nucleic acid recombinant and protein also another one. Suppose when you are joined with the one DNA and another one, sample one, two DNA, recombinant, as a result, the one protein molecule secreted. That protein molecule is called as recombinant protein vaccine. It is also fight against many diseases. And also it has a property for vaccination property. So one. Vectored vaccine, vectored. And inactivated vaccine and live attenuated vaccine. These are the types of vaccine. See, these are the things that are given. Okay, so this is very, very important. Okay, for your future. Okay, see, this is the vaccine development history. It cannot be clearly visible. Okay, those are giving 1796 onwards and in 1954. Here 207 only. Okay, I have taken. So next one we can see it fully. In 1796 onwards, we'll get up to the level of 21. Okay, the COVID vaccine up to the level of COVID vaccines is 2021 and up to the level of until the 2021 I have taken here. It will be clear means you have to go through it. Okay, it will not clear because it started from the 1796. Okay, and here this one is called as Jenna. Okay, 1796. So from this one, see how many years. See that last one up to this level. Okay, so the history of the vaccines. Okay. Go to the next one. So first generation. Next we will pass on to the another things. The vaccine is also prepared. Now which generation we are present? Vaccination generation. Will you please anybody else? Which generation we are all are living? We cannot able to find out, isn't it? first generation, second generation, third generation, fourth generation, fifth generation, sixth generation, like this, it's going on. See the first generation and the second generations. See, this is the first generations. Okay, and the nucleoprotein, this is the normal one. How it will be like this? This is the second generation of this one. Live attenuated vaccine. This one is inactivated vaccine, and also like particle vaccine. This is also a second generation. This is the inactivated. This one is also a second generation. How it will be? And live attenuated vaccines. Live attenuated vaccine is also converted to which one, you know, vector vaccine. This is called as first generation, and also this one is called as second generation. This one is called as third generation. Okay, and these are the third generation. See here, these are all a second and a third generation. These are all our first and second generation. Here, so these are the developmental stage of the generation wise. Okay, the vaccine, how they were prepared. Initial stage, the microorganism will be like this. After the third generation, the microorganism will be like this. It will be attenuated. Okay, third generation will be like this. These are the developmental stage of the vaccine preparations in the laboratory aspect. Okay, see the first generation attenuated microorganisms. The second generation inactivated the microorganisms. Third generation, see that toxoid polysaccharide, split plasma derived polysaccharide and conjugate. These are all isolated from the inactivated microorganism. Then it will be used for the preparation of vaccination for the particular diseases. Fourth generation, subunit of by recombinant DNA technology. Recombinant means here there is no any, what is the nucleic acid. The third generation, only the component only they have derived from the microorganism, isn't it? After that, DNA is a very dangerous one. RNA is also very dangerous. We took from the DNA and used for the vaccination process, for the technological aspects. Okay, what are they? Hepatitis B and hepatitis virus. Okay, and fifth generation, reverse vaccinology, AIDS, cancer, dengue fever, and malaria. These are all what is that? Generation aspect they have splitted or classified the vaccination process. First, attenuated. Second one, inactivated microorganism. Third one, toxoid polysaccharide, split plasma derived polysaccharide and conjugated. Fourth one, the DNA and RNA molecules. The fifth one, reverse vaccinology. They have given the what is that? Target diseases. Okay, the influenza, measles, mumps, polio, rabies, tetanus. Here also given, isn't it? Influenza, pertussis, plague, and polio, typhoid. The diphtheria, influences, hemophilus, influenza, hepatitis B virus, pneumonia, tetanus, typhoid. Here, hepatitis B and hepatitis virus. The AIDS, cancer, dengue fever. These are all advanced level, isn't it? These are all primitive stages. This one is called as advanced level. Okay, so this is very, very important. See the type of vaccination, first generation vaccine, the vaccination are prepared by the inactivating the whole pathogen. Note it down. Inactivated the whole pathogen, but they have a side effects. Note it down. Oral polio vaccine, OPV, DPD, diphtheria, pertussis, tetanus. Okay, next second generation vaccine, how it will be? Antigenic polypeptide of the pathogen are produced with recombinant DNA technology in transgenic organisms. This is another thing, transgenic organism. It is also prepared from the combination. DNA technology means, what does it mean now? How will you prepare humulin? Human insulin, how will you prepare in the commercial purpose means you have to use the DNA molecules of the particular polypeptide. You have to cut the nucleotide from the particular donor, isn't it? That is called as molecular scissor. After that, it's also connected with the host DNA with the help of the ligase enzyme, DNA ligase. Then introduced in the E. coli, it will be produced a new copy. Okay, that is called as genetic engineering or recombinant DNA technology. These are made by multiplication of surface antigen by genetic engineering. Already told you, they have no side effect. Example, hepatitis B vaccine produced from the transgenic yeast. This is very, very, but transgenic, transformation of genetic material from where to where, yeast to human beings or virus or like that. This is called a second generation. Third generation, these are highly potent and synthetic in nature, prepared by gene. They are all called as DNA vaccine. Third generation vaccine is also called as DNA vaccine. Okay, so first generation, second generation, third generation. Already we discussed the previous tabulation up to the fifth generation, isn't it? See here, the vaccination program for the 0 to 5 year baby, how it will be? What are the things? This is also very, very important. So many times they were asked the question. And the infants, after the birth, at the birth, the first vaccination, which one of the following? Like that, they may ask the questions. BCG is the first one. BCG is the first one. Oral polio vaccine, hepatitis B1 only have to give. 1.5 months, we will provide a diphtheria and pertussis tetanus 1, oral polio vaccine 1, hepatitis 1 again, hepatitis B2, B denoted as, which one you know about. And 2.5 months, TPD 2. This one is 1, this one is 2, this one is 3, diphtheria. Here 1, 2, 3, hepatitis 1, 2, 3. So likewise, 2.5, 1.5, 2.5, 3.5. You have to provide. This is the important. After 6 months, hepatitis B3 only. 9 months, measles. 15 to 18 months, DPT. Okay, and also which one the booster MMR. And 4 to 5 diphtheria again, DPT. Second booster. You have to provide. These are the steps. These are the format. Already we also, what is that? Attenuated by this type of vaccination process. In our body also received the vaccination like this. See this vaccination for 1796 smallpox, 1880 rabies. Earlier, see this one is called as introduced diphtheria, toxoid vaccination, tetanus and diphtheria. And polio vaccine 1950, hemophilus influenza 1985, hepatitis A 1991, retrovirus 1998 only. Okay, and also HPV vaccines. This one 206, Ebola vaccine 2019, COVID vaccine 2020. These are all are isolated from which collected from the Google. It is not present in your book. Okay, just for the information, just note it down. And next we'll pass on to the disorder. We will finish it up to this level only, I think. Immune system disorder. What are the type of immune system disorder? When it will happen? Improper functions of the immune system may cause discomfort, allergy, disease, even death, anaphylactic shock. Very about immune response cells, immune system, immune organ system. They can say beta lymphocytes, bone marrow, thymus glands, and also lymph node, lymphoid organs. Okay, these are all are produced the antibodies, isn't it? Suppose it produces more number, some other chemical substances, some other another components, physiological or biological or chemical substances involved enter in the body. As a result, it damages the cells. Okay, it damages the cells. As a result, what happened? It will not produce the diseases. So that is also an immune system disorder. Okay, they may be divided into 3 classes: hypersensitive disorder, allergy, autoimmune disorder, and immunodeficiency disorder. These are the 3 types of diseases. The 3 types of improper functioning of the immune system have been reported by the many scientists. Okay, see, and hypersensitive disorder, what are they? Physiological allergy, bronchial asthma, hay fever, anaphylactic shock, and eczema. These are the hypersensitive disorder allergy had happened. Yeah. Why?
So what is anaphylactic shock, sir?
Anaphylactic shock. Okay, that 2 things I didn't prepare for this one. I will tell you. Anaphylactic shock. Okay, so I will tell you. Okay, where is it? Anaphylactic, isn't it? Yes. See, anaphylactic shock, isn't it, Ba? Yeah.
Yes, sir.
Yeah, see here, anaphylactic causes immune. Can you see this here? Can you see this one?
No, sir.
No, okay. So I will tell you, anaphylaxis causes the immune system to release a flood of chemicals that causes you to go into shock. Blood pressure drops suddenly and the air narrow, blocking breathing. Signs and symptoms include a rapid, weak pulse and skin rashes and nausea and vomiting. This is called as what is that? Anaphylaxis. Okay, a shock. Suddenly it happens. Blood pressure drops suddenly and the airways narrow. That is called as blocking of breathing. Breathing also, it will be affected, isn't it? These are the two things. Another one is BCG, isn't it? BCG you are asked. What is the full name of BCG, isn't it? BCG.
Yes, sir.
Yeah. So, full form, isn't it? Yes, full form. Sir, see this here. Bacillus calamity gowrin. Note it down. BCG, Bacillus, BACILLE. Okay. See, I will tell you. See here. Sorry. Bacillus calamity gowrin. Already saw it, the things. See here. Okay, see here. One second. Bacillus calamity, and I will tell you. BCG, isn't it? See here. Okay. Yeah. Okay. See here. Okay. I will. You note it down, pa. Bacillus calamity gowrin. You got it?
Yes, sir.
Yeah, got it. Okay. So today what we have seen, so what is by artificial immunity? Okay. What is by natural immunity and artificial immunity? That things already we discussed. Types of acute immunity, active and natural. The active immunity also further divided into two types. That is called as natural and artificial. Natural immunity already present in your body that fight against many diseases such as smallpox, measles, mumps, and also natural active immunity is also present. That means a natural immunity organ system present in our body that fight against many diseases naturally. That's why it is called as natural active immunity. Artificial immunity is nothing but the what is that? These are the artificial. Artificial immunity nothing but attenuated from outside. Preparation of live or killed vaccine of the microorganism, killed by the microorganism and also used for the immunization process. That is already given here, lived and killed. Okay, these are the things. BCG, Bacillus, what is that? Calamity gowrin. Okay, MMR already, TAP. Okay, tetanus, antibacterial vaccines, enteric fever. I will tell you that things also. So this is a preparation, already we saw it, that things also. And next one is passive immunity. Passive immunity is nothing but what is the name? The ready-made antibody we are giving to the particular person. As a result, it will induce the inner side. It is also further divided into two types already but natural and also artificial. Natural means already will be immunoglobulin responsible for the natural passive immunity. Particularly immunoglobulin G is a antibody. They can cross from the mother to fetus through the way of placenta. And mother milks also, which one in the colostrum, it contain more amount of immunoglobulin G. Okay, normal milks already colostrum is also about this one. Colostrum is the first milk, is a container more immunity power compared to the normal milk. Okay, the colostrum is rich containing immunoglobulin A antibodies already about that things also. Artificial passive immunity. Artificial passive means also another type of immunology, and passively transferred to the recipient by the administration of the antibodies. This is also hyperimmune sero obtained from the animals or any other mammals. How it will be already well about anti-rabies, diphtheria, and also tetanus serum also isolated from the horse. Not only the horse, chimpanzee, monkey, mammals particularly, because we are all around mammals. That's why. And primary and secondary immune response. When it gives a, when it the first exposure of the antigen, it is called as first, and suppose if it is the second time, it means a second. Okay, that is called as immunological memory. The primary immune response, it will be very less compared to the second times. It is a more double than that of the first one, previous one. Like that already we saw it in the graphical representation, isn't it? And then vaccination. What is meant by vaccination? Who described vaccination? What are the types of vaccinations? Okay, and who is the father of vaccinations? That things also already we saw it, isn't it? Types of vaccination already we discussed, and also and how they will prepare DNA and who is the father. All those things already we discussed. And then generation aspect, first generation vaccine, second generation, third generation, up to the fifth generation. Why up to fifth generation we will discuss in this. This is the recent one. Sixth, seventh also having. Okay, then already know the first generation, second generation, third generation. First generation, what is that? Second generation, what is that? Third generation, it is a combination of the DNA. When the DNA is used, that is the third generation. This is also so many times they were asked, which generation DNA molecules, DNA is used for the vaccination process, like that. This is very, very important. Don't forget it. Okay, this is also very, very important thing. And then immune system disorder, just we discuss. Remaining things we will see on the next classes. Will you please, if you have any doubt, please ask me. Anaphylactic shock, already we discussed. Sudden changes occurs. Okay, sudden changes mean breathing as well as a blood clotting, blood also. Okay, that's a discomfort. That things will lead to death finally. Okay, my dear students, that's all about today class. We will see the remaining on the next class. Okay.
Thank you, sir.
Thank you, Ba. Thank you. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Thank you.